So the search for genetic material. So transforming principle was given by the Griffith, and he worked on Streptococcus pneumonia bacteria, and the biochemical characterization of that transforming principle was given by Avery, MacLeod, and McCarthy, and unequivocal proof that DNA is the genetic material is given by Hershey and Chase experiment. Right now, in transforming principle, which is given by like Griffith, Frederick. um so there are like S colonies and R colonies. S colonies, these are virulent, they have protein code, and R colonies do not have any protein code, so they are non-virulent. Okay, so at the fourth step, at the fourth stage, when he like S strain heat killed and R strain, he mixed it and injected into the mice. Now something has been passed from heat killed S strain to R strain, and R strain get transformed into the virulent, and that kills the mice. So this is it. He concluded that the R strain bacteria had somehow been transformed by heat killed S strain bacteria. So some transforming principle transfer from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. Okay. Now the biochemical nature of the chemical genetic material was not defined from this experiment. So now the biochemical characterization of transforming principle. Savary, MacLeod, and McCarthy, right? What they did, so they purified biochemicals, that is proteins, DNA, and RNA from heat-killed S cells to see which ones could transform live cells into S cells. So now you see, like they discovered that a protein digesting enzyme, that is protease, and RNA digesting enzyme, that is RNase, did not affect this transformation. So the transforming substance was not a protein nor or so it wasn't a protein nor RNA. So digestion with DNAs did inhibit transformation, so suggested that DNA caused the transformation. So they concluded that DNA is a hereditary material, but not all biologists were convinced. Okay, and DNAs are different. So genetic material is DNA. So the unequivocal proof that DNA is the genetic material came from the experiment of Alfred Hershey and Martha Chase, that is in 1952. They work with viruses that infect bacteria called bacteriophages. So they infect the E. coli cell with the bacteriophage viruses. Okay, so this is it. So this is all they did, right? Now they grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. So viruses grown in the presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Okay. Similarly, viruses grown on radioactive sulfurs contain radioactive protein but not radioactive DNA, because DNA does not contain the sulfur. Okay. So when the radioactive phages were allowed to attach to E. coli bacteria, then as the infection proceeded, the viral coats were removed from the bacteria by heating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Okay. Now I just see in here. So no, using the bacteriophage, that is radioactive sulfur S35 label protein capsule, and radioactive P32, that is radioactive like phosphorus label DNA. So when the infection, blending, and centrifugation occurs, you see at the first case on the left hand side that no radioactivity 35 sulfur detected in cells, and radioactivity 35 detected in supernatant. That is protein code is removed. So this is not transferred in the like the cell. But when the radioactive, that is P37, detected inside the cells, that means something has been passed from the outside to the inside. So no radioactivity detected in supernatant. So the properties of the genetic material, that is DNA versus viruses. Okay. Now you see, a molecule that can act as a genetic material must fulfill the following criteria. It should be able to generate its replica, that is replication. It should be stable chemically and structurally. It should provide the scope for slow changes, that is mutation, that are required for evolution. It should be able to express itself in the form of Mendelian characters. Okay. Now both the nucleic acids show the complementarity. Okay. Griffith's transforming principle also tells the stability of the genetic material. So 2' OH group present at every nucleotide in RNA is a reactive group and makes RNA liable and easily degradable. So RNA known as to catalytic, that hence reactive. So DNA chemically is less reactive and structurally more stable when compared to RNA. Also thymine provides the additional stability to DNA, right? Now both DNA and RNA, they are able to mutate. In fact, RNA being unstable mutate at a faster rate, and consequently viruses having RNA genome having shorter lifespan mutate and evolve faster. So RNA can directly code for synthesis of proteins, hence can easily express the characters. So RNA can express the characters, while DNA being dependent on the RNA synthesis and then the protein. So as you can see, the protein synthesis machinery has evolved around RNA. Important line. Okay. Now both DNA and RNA can function as genetic material, but DNA being more stable, so for prepared for storage of genetic information, for transmission of genetic that information is the RNA is the better. So RNA world now. RNA world, right? The first genetic material. RNA was the first genetic material. Even though the genetic material is DNA, but RNA is the first genetic material, right? There is now enough evidence to suggest that the essential life processes such as metabolism, translation, splicing evolved around RNA. Okay, so all these things has evolved around RNA, right? RNA used to act as genetic material as well as catalyst. But RNA being a catalyst was reactive and hence unstable. Its catalyst is reactive and hence it is unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. So DNA being double stranded and having complementary strand for the resist changes by evolving a process of repair. So replication given by like Watson and Crick. Double helical model was also given by the Watson and Crick, right? Now base pairing also given by the Watson and Crick, right? Now they also give the replication in 1953. Now what this model suggests, the two strands would separate and act as template. So both the strands act as template for the synthesis of new complementary strands. Now after the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. So this scheme was termed as like semi-conservative DNA replication. Okay, you see Watson-Crick model for semi-conservative DNA replication. Now experimental proof. Now this the DNA replicates semi-conservatively. Experiment on like E. coli and then on the Vicia faba, that is higher plants, by the Taylor using the radioactive thymidine, and on E. coli you see there is heavy isotopes, that is N50. First they grew the E. coli in a medium containing N50, that is heavy isotope. Okay, so once the isotopes get incorporated into their DNA, then they place it on like the N40 medium. Okay, so after first generation, then second generation, then third generation. Okay, so an E. coli divides in like every 20 minutes. And this was given by the Michelson and Stahl. Michelson and Stahl. So Matthew Michelson and Franklin Stahl. Okay. And N50 is not a radioisotope; it is a heavy isotope, as I told you. Like now, as you can see. So after 20 minutes, there is like hybrid DNA. So N50 and N40, because it is placed in the N40 environment. Then and again when it is placed in N40 environment, so after 20 and 20, so after like 40 generations, so after like 2 generations, so it's light and hybrid. Okay, so from heavy DNA it's getting into like hybrid and then light and light, so hybrid then light. Okay, so the quantity of the light is getting increased. Okay, so that's all light. And remember that is after 20 minutes. E. coli divides in 20 minutes, right? Had a hybrid or intermediate type. The DNA extracted from the culture after another generation, that is after like 40 minutes. And second generation was composed of like equal amounts of like equal amounts of these hybrid DNA and light DNA. So after like third generation, you can see like 3:1, that is light are 3 and 6, then hybrids are like 2, so it means like 3:1. So as you can see, ncert is also asking like if E. coli was allowed to grow for 80 minutes, then what would be the proportion of light and hybrid densities DNA molecule, as you can see. And okay, the experiments prove that DNA in chromosomes also replicates semi-continuously. So DNA in chromosome also replicates semi-consecutively. So the machinery and the enzymes. Okay.